we have Adrien Roux coming. He holds a PhD in biology from the University of Geneva and has worked in different companies like Thermo Fisher and Merck Serono. Since 2019, he was nominated as an associate professor of bioengineering and biology at HEPIA HES-SO in Geneva at the University of Applied Sciences in Geneva. And he's working now at the campus Biotech, where he focuses on applied research and industrial applications to develop and optimize complex in vitro cellular models for replacement of animal studies.
So good afternoon, everyone. And indeed, today I will try to show how engineer can help us as a biologist to develop novel replacement model, and more specifically in neuroscience. And I have decided to focus the talk on experience that I have in the lab, but specifically with companies. Why companies? It's just because I think that they are part of the ecosystem and they can also contribute to make an impact in the 3R. So as Marcos said, I'm a University of Applied Science, so I'm an academic, but I'm really focusing on applied science. And my bioengineering group is composed of both biologists and engineers. So if you want, the biologists are really the end user. They are this is the people who say, I would like to do this in vitro model and so on. And so they start to have some needs, and we need some people to help us. And this is the engineers, the solution makers. We are based in Geneva, the nice campus Biotech. And our biologists have their own expertise. So they are doing complex in vitro cellular models. And when I say complex, it's both for the 3D neural tissue, or organoid, and so on. And sometimes it's also co-culture. And co-culture with 3 or 4 types of cells, it could be quite complicated. Since 8 years now, the lab has completely switched from embryonic stem cells to human iPS-derived cells. And sometimes, indeed, within this in vitro model, we also use other type of samples like plasma or extracellular vesicles. If our specialty is really neuroscience, and we do have also other type of in vitro model in our lab, because when you are expert in the barriers, more or less all the same react the same. And so this is why we are working also on the lung barrier, intestinal barrier, and we have a very nice project on endometriosis with endometrium. In our laboratory, we have decided to focus on long-term culture, long-term effects. So it could be for neurotoxicity or to see the drug discovery and so on. And for this, we need specific readouts. We do think that indeed gene expression is a very nice readout, but having a functional readout is really important. And for neurons, the functional readout is the electrophysiology. So the capacity to have an electrical signals. For the blood-brain barrier, the functional readout is really the capacity to maintain this barrier. And we will also try to monitor this barrier with specific techniques. And our engineers have also their expertise. And first, I think they are able to do fast prototyping techniques. So indeed, now everyone has a 3D printer in his lab, but not everyone has some engineer who are really able to understand how to design microphysiological systems and all this microfluidic channel. And also we can use it by either additive or subtracting methods, which are not the same for this. And then we can implement plenty of other techniques like fluidics, optics, electronics, and so on. Electronics is really important because it's the link between all the technology to control a camera, to control a sensor, to record everything, and it's really important. And we have also the chance to have in-house a specialist in microfabrication, so we are able to design and to build our own electrodes here in the clean room, and this is how we can do this organochip. So really, in our lab, the possibility to have both the biology and the engineering together make that it's easier for us to make a model. But let's refocus now on really the replacement model. Where should we start? We should always start from the need. So the idea is really, and the need from who? From you, the biologist. So you can work in a public institution, a research institute, or even a company, but we need to start from your needs. Meaning that the first step is really to understand what you need and what you don't need, because otherwise you can have a lot of nice to have in your organochip, but this is not the aim. And then after, when you have all the needs, we start to write all the specifications, the technical specification, and from this we say, what are the possible solutions? Here also in our group, the idea is not to reinvent the wheel. So if there is already a solution, we'll try to implement the solution, even if developed by another team or by companies. And then after, we start the process of designing and prototyping and pre-validating, and we can restart this process several times before this pre-validation step, where we stop everything, we do design freeze, as we call it, and then we start also the transfer and the dissemination. The transfer is the very important part, because we are not developing the organochip only for our lab. The idea is to make an impact, to have these models available everywhere. You know now that with the HEPIA cells, everyone could start from a different material, a different patient. So it's very complicated to reproduce the data. So at least we need to have common tools, and this we can disseminate all these technologies. So how to answer the needs? So we have our toolbox, and with this, I told you already about all these different points, and this is the way to go from our concept. So our concept always starts by drawing on a computer how it will look like. And to be honest, when we are at this stage, some people are already very happy, but the most difficult part is to switch to the reality. And when we switch to the reality, we start already to see some problems sometimes. Here you can see some examples where we do have some interconnected wells or different membranes, some electrodes, and so on. But the aim is really to go up to the validation, which is the most difficult to achieve. And when you have validated your technology, you can go to technology transfer, and sometimes it can become a product. You see also that there is some screenshot from, and this is really important, you know, because all these pump and electrovalve are controlled by electronics, and these electronics have firmware inside, but also software. And it's really important to understand that we, as a biologist, we don't want to code or something like this. We want something really simple, and we want a user interface quite easy to work with. So it's also an important point for this. But let's going back to the story, where I give you 4 examples with 4 companies. The first company is based in Geneva, and basically they are specialized. It's a small pharma company specialized in glial cells, and they have a really nice model, in vivo model, in vitro model, but they were using 2D in vitro model, and for them it was not enough. So they didn't have the technology to work in 3D. They asked us if we can make a collaboration. We were lucky enough to have a grant to work with this. And so we have started to develop a 3D in vitro model for them with their cells. which are really specific. And then we have adapt also all their readouts, because all their readouts were initially adapted for 2D, but imaging we need to switch to 3D, and more specifically for the microelectrode arrays, the electrophysiology. So at the end, it looked like this, where we have characterized the model, the cells. In this case, it was a neuronal cells with some glial cells also. Then we are able also to work and to look for calcium imaging. But I will go, I'm already a bit late, to be honest. So I will try to go. This is what you can see on the right, but we can also stimulate the cells if it's needed. In our case, if the neurons are mature enough, it's not needed to really bring an external stimulation. But what is the output for this company, GliaPharm? The output is the cellular model. So at the end, they have a 3D model, so they were happy about that. They do have also a custom platform. In their case, the initial needs were to have at least 4 reservoirs. One was for the cell culture medium, and 3 of them were for different concentration of drugs. Then we had electrovalve. We have also all the electronics for the electrophysiology, plus a camera for recording the video behind. And so this platform is now in the laboratory, and they were also using this to publish some paper where they were very kind to put us as co-authors. Then the second company is Multi-Channel System. It's a German company, quite well known in the electrophysiology field. So they have already plenty of setup, plenty of instruments, but it's also very well dedicated to the 2D cells. So basically, the cells, the neurons are laid and attached on the glass with some electrodes. And now we have seen it in the 2 first talk, are working more and more with 3D, but we need good tools for that. So they ask us if we can develop for them a new MEA, multi-electrode array biochip, but fitting in their existing instrument. So this is what we did. So here in this case, we have 60 electrodes with standard 3D connectors, so Lure. So you can use your own Friedic system, and on top of that, you can put your tissue. So your tissue can be cerebral organoid, neurosphere, hippocampus slices, or assembloids. We have tested everything. You can see it on this paper here, and that was really the point, to not change the instrument. The third company is also based in Geneva, Switzerland. So they are working on blood-brain barrier, and more specifically on leukocyte trafficking. So at the moment, they have a setup where they are looking for leukocyte trafficking through a single chamber system. And so they are CRO, contract research organization. So their clients are big pharma, and now they are asking for more complex model with dual chamber system. And so they came to us to say, okay, are you able to develop a novel microphysiological systems with 2 chambers instead of 1, and with more high throughputs? So, but that's really important to say that they have started first to look at what is already on the market. They have tested 5 biochip microphysiological system. I will not quote the brand, but they have tested 5 of them, and none of them were good enough for their model. So that this is the reason why they have asked us to develop their own system. You can see that indeed there is 2 compartments here. You can still do the leukocyte trafficking, and we do have on the other side also some neuronal cells. So we have increased the complexity also by adding neuronal cells, and before it was only blood-brain barrier cells. So unfortunately, I cannot put the real picture here because they will patent the device, but I can just tell you that it's really between the 2 compartments, it's a porous membrane, and the cells are able to communicate from one compartment to the other, and also to do transmigration. And maybe it's maybe more for the Swiss, but I think it will be also an interest for the French. It's all this is as a cost, you know, and so and we are academics, and so there is always possibility to find financement. And in our case, it was through InnoSwiss or something like this. Sometimes we do direct contract, and at the end we had a big grant to try to help the company to develop their business. And we do also other type of stuff, such as here, where it's a European project, with 50 partners. It's really a lot, but the aim here is to develop technologies for different companies within Europe. In this case, we have developed a device to monitor the transendothelial electrical resistance, called TER. This is really the bioimpedance. So we have developed all the electronics, all the hardware, all the electrodes to fit exactly in the already existing device from the company Alveolix, like this. So this was something for them. And then we have also started to work. So this is not achieved yet, but this is a concept that we have, where instead of having 2 electrodes in liquid, 1 electrode will stay in the liquid and the other will be at the air-liquid interface. So this is quite important because when you are working with the lung barrier, for example, they are always at the air-liquid interface, and we can see that to maintain a good oxygenation, some in vitro models are also at the air-liquid interface models. So I'm just finishing on time, just to explain that I strongly believe, as a biologist, that I need the engineer and the supporting technologies to go further in the replacement in vitro model for this. But for me, what is really important is to understand that even for technology, we need really specific model. You know, for the biology, it's quite clear. Now we are talking about personalized medicine, but I think that we need also personalized technology for our in vitro model. With this, I just thanks again my team, and I'm open for some questions.
Thank you very much, Adrien, for this nice overview of what you are doing and what kind of collaborations you have. We have time for a few questions because you were good in time. Mm-hmm. Thank you very much for adhering to the timeline. So are there some questions from the chat?
Yep. Thank you, Adrien, for a nice talk. You showed many devices which you designed now. What would you think as a timeline when these are broadly available and how user-friendly they will be?
So it's a good question. First, we are not starting from scratch. We have always what I call the toolbox, meaning for the bioimpedance, we have now finished to develop the electronics device. So it's more adaptation and implementation. But for difficult project, it took 18 months full time to develop something. For easy project, some of them it was more 6 months. And when you want to go up to a product, it's a bit different because for GliaPharm, they keep the device in their lab, and they use it as a research for alveolics or for multi-channel systems. They want to sell this as a product, so you need to add an extra year on top of that.
Okay, that's quite fast, I would say. Thank you. And then there was an interesting question as well. You use always machines to measure the readout from your brain organoids. Could you also think about genetically encoded voltage indicators?
It's a good question. Sometimes we don't use this idea. It's a good idea, to be honest, but we use calcium imaging. So sometimes we don't record the electrical activities, but we just put a camera and some calcium dye, as we saw just in the presentation before. And with the calcium dye, we are able also to see some functional activity. So I think that calcium imaging for neuroscience can be complementary to electrophysiology. But I just want to add that electrophysiology, the bottleneck is really the data analysis.
Okay, thank you. Do we have more questions, or is it we go on, Markus?
If we have another question, that would be fine. Do we have one? Or
yeah, there was another question. It was more about the endothelial cells in your system. Are they growing under fluid stress?
Yeah,
because it infects the tightness.
Yeah, indeed. If you want a good tight junction and TER, adding a shear stress is adding relevance to your model, and this is the case. And I will also, because I do see the chat also, I will answer one question. Yes, our organoids do have also some myelinization, because we are able to keep our neurosphere, brain organoid, and so on for a long period of time. Some of them in the lab here are 2 years old now. And so after 3 months, you start to see some myelinization.
Okay, good. So thank you very much again also for these vivid discussions, Adrien. It's always a pleasure to see you.